Okay, we're going to get started here. So thank you all for joining. We wanted to welcome everyone and thank you for joining us. I'm Glenn O'Neill, president of Cure Sanfilippo Foundation, and we're pleased to be collaborating with the National MPS Society and Ultragenyx to bring you this event. The efforts to bring about this gene therapy for Sanfilippo type A have been ongoing for well over a decade now, beginning in the labs of Dr. Fu and Dr. McCarty at Nationwide Children's Hospital. Also, the adjacent natural history study was conducted at Nationwide Children's Hospital, and then that led to the initiation of a clinical trial in 2016. Those steps would not have been possible without funding and support from many patient advocacy organizations along the way. During our 90 minutes together, Ultragenyx will provide our Sanfilippo community with an update on this clinical trial program, what has been learned to date, and the path forward. Thank you to everyone who submitted such thoughtful questions. These questions have informed the presentation that you will hear today. Of note, the event is being recorded and will be made available for later viewing. Also of note, we want to make sure you know that this event was organized in response to the questions from the Sanfilippo community and is intended for audiences in the US. The safety and efficacy of UX111 have not been established or approved by any regulatory agency. Now it's my pleasure to introduce you to our speakers and moderators for today's event. Leading the UX111 program at Ultragenyx, we are happy to welcome Dr. Heather Lau and Kristen Voorhees. Dr. Lau is a pediatric neurologist and the executive director of Global Clinical Development. Kristen Voorhees serves as a senior director of patient advocacy, leading patient engagement for Sanfilippo syndrome type A. Moderating the Q&A portions today, we have Dr. Matthew Ellinwood, the Chief Science Officer at the National MPS Society, and Dr. Cara O'Neill, the Chief Science Officer and co-founder of Cure Sanfilippo Foundation. Thank you again, everyone, for your interest and your attention. We have a lot to get through, so we're going to get going. With that, I will hand it over to you, Kristen.
Thank you very much, Glenn. Hi, everybody. Let me just share my screen. We're going to switch here. So good morning and good afternoon, everybody. As Glenn said, my name is Kristen Voorhees, and I'm really honored to be here today. I'm joined by Heather Lau. She'll introduce herself in a minute. I just want to thank both the National MPS Society and Cure Sanfilippo for being such great partners and advocates and really strong champions over the years, and we're excited to be able to share this information with you. I'm Kristen Voorhees. I've been with Ultragenyx for about 6.5 years, and again, I'm really honored to be here. We have a lot to cover with you, so we'll keep the introduction short. But Heather, would you like to say a word?
Sure. My name's Heather Lau, and as you heard, I'm Executive Director of Global Clinical Development. But previously, prior to joining Ultragenyx 3 years ago, I had run the lysosomal storage program disorder, lysosomal storage disorder program at NYU for 10 years, where I was conducting clinical trials in protein-based, small molecule, and gene therapies. And I'm happy to be here at Ultragenyx leading the MPS 3A program, and I'll be talking about our recent update today. Thanks.
So as Glenn mentioned, and Heather as well, our objective today is really quite simple. We're here to discuss updates to the program that were shared as recently as last month at World Symposium in San Diego. And we received about 60 questions from the global community, and all of that has helped to not only determine what Heather is going to present, but we've also organized those questions so that Matthew and Cara are going to be moderating, and we'll explain a little bit about how that's going to work. So this is our agenda. We originally scheduled this for 75 minutes, and thankfully we're able to pull it together for 90 minutes. So thank you to everyone who has signed on and will be listening to this later. We do have a lot to get through, and so I would just recommend that because it's going to be recorded, come back and listen to it. You'll be able to pause and maybe digest the information a little bit more. In case you're not familiar with Ultragenyx, we'll spend a minute or two just setting that stage. We're going to talk about how we're able to communicate with this community. We're going to talk about a little bit of information and context about how we can respond to your questions today. And then Heather's going to do a brief background on the gene therapy program for MPS 3A. We've then divided the sections into two main ones. The first, we're going to spend time talking about the common challenges in developing treatments for MPS 3A and important issues in all gene therapy research. We'll take a break for a few minutes with a Q&A that Matthew will kick off with about dosing and redosing. So those will be responses to your questions. We'll then go back to Heather for more of a presentation on the preliminary results from the UX111 Transfer A and long-term follow-up studies. We'll then take another few minutes for a Q&A, and the topics we'll cover then with Matthew and Cara will be efficacy, safety and adverse events, and the future of the program. And then there were some other questions that we received that really didn't fit into these categories, and so we'll spend time addressing those at the end. I have a feeling that many of you already know who Ultragenyx is due to our founder and CEO, Dr. Emil Kakkis, but in case you're not, we were founded in 2010. Emil has a very strong and long-standing background in MPS, and he founded Ultragenyx to really create a steady pipeline of new and effective therapies and to be able to bring them to market for the treatment of both rare and ultra-rare diseases. We're headquartered in Northern California, which is where I am today, but we're global and we have more than 1,200 employees. The picture to the right is from about a year and a half ago when we were all able to come together in person. And so we've really grown a lot over the years. And as part of our commitment to patient-focused drug development and communities, we're really here to work with you in deep and meaningful ways to be able to understand what's important to you and address them both through education, information, but of course through advancing new treatments. So I want to spend a minute talking about our commitment to the community in terms of how we're able to communicate. We're really grateful to our partners at the Society and Cure Sanfilippo today that they are recording the presentation and they're going to have a copy of that recording. And this presentation and town hall is really a follow-up to a community update we shared last month following Heather's presentation in San Diego at World, where we were able to present on the preliminary results of the UX111 program. And so at that time, we provided the community organizations with written letters announcing that news. And that's really one way that we're able to stay in touch with you. And so it's up to the advocacy groups to be able to share that with the communities as they wish. And we're grateful that we're able to have that strong partnership and path. I also want to point out that there's a number of ways that companies like ours can communicate about different study results. So we're committed to sharing all of this information, both positive, negative news, when it is, in a variety of ways, and that includes posters at conferences and congresses, presentations, and then in journal manuscripts. And so we do submit the results of all of our sponsored trials for publication in peer-reviewed journals and at meetings. And after a study is over, so about 1 year after the last patient in the trial has had their final visit and a formal study report is put together, that then allows us to prepare plain language summaries for all of our programs. So for example, about a year after the last patient in the in the ABT-001, the Transfer A and the long-term follow-up studies are each completed, we'll be able to put those together. And so then those summaries are shared with both the trial participants through the investigators, and we also share them on our website at ultragenyx.com. It does take time, and I think that that's one of the reasons why we're here today, is to be able to talk about what we know in an interim fashion. As we mentioned, we received many, many thoughtful questions from the global community. We're so thankful for that. We received about 60, and so you'll see in a minute that we've organized those by theme. And I think we've already, acknowledge that while we can't take live questions today, please feel free to pass them on to us so that they can be addressed later. Okay, so right now you're going to see about 3 or 4 slides that might seem really busy. So I want to orient you to that before I pass it over to Heather. So we had about 8 to 10 themes. So you can see here on this slide, there's one about efficacy, there's one about early dose cohorts, another about eligibility criteria, and so on. And so in each section of the presentation today, what we've done is either it's directly informed Heather's talk, which means that those responses to these questions have already been incorporated. And the questions in bold are going to be part of the moderated Q&A with Heather and, with Cara and Matthew. So for example, we also have themes of safety and adverse events or effects. We have immune response. The future of the program is probably the largest category of questions, and so we're going to talk about that at the end, namely through the moderated Q&A. And so this is about when might it be approved? Can you share timelines? What are your next steps? And then finally, there were some others that didn't really fit into the previous categories. So you're asking about, is there an application of study outcomes for Sanfilippo type B? How were patients in cohort 3 of the transfer A recruited? And how is this different from the gene therapy product that is in development at a certain institution? So at this point, I'm going to hand it over to Heather, and she's going to kick us off, and then Matthew and Cara will come in at certain points to moderate the Q&A. Thank you so much for being here.
Thank you, Kristen, and thank you to both Cure Sanfilippo and the MPS Society. It's a pleasure to be here and talk to you about our recent data sharing. So the background to today's presentation is that we are going to provide information from the ABT-001 clinical trial from our most recent data cut from August 16th, 2023. And this was presented at the World Symposium on February 9th, 2024, and then again at a workshop that I'll mention later regarding CSF heparan sulfate. Now, it's important to understand what this data cut was focused on. It was on at least 1-year follow-up of our 17 patients in this modified intention to treat group, which is part of our cohort 3 or highest dose. And I'll be referring to this cohort throughout the presentation. And this group was created or formed prior to our transition as sponsor. And this group, also called target population, is essentially our registrational cohort in the phase 3 portion of our ABT-001 study. And I will get to that in more detail in later slides. Today, we are unable to discuss data or disclose other information that has not been already published or shared publicly, as it may be currently in peer review with study investigators and may not be shared due to restrictions set by scientific journals or medical congresses. Next slide. Now, as you saw, there's many different questions that we are trying to address throughout this presentation, and we've received several questions about cohorts 1 and 2, and these are our early dose cohorts. And I do want to point the public to the available information that was presented at the ASGCT, or the American Society for Gene and Cell Therapy Conference in 2022. And that information was presented by Dr. Kevin Flanagan, one of our PIs, and that went into data associated with cohorts 1, 2, and 3. Now, as of today, we are continuing to analyze and interpret data from all cohorts, and that's actively in progress because we follow these patients even though they might have completed that initial study in cohort 1 and 2 in ABT-001. These patients are followed in a long-term follow-up study. And it takes significant time and resources, even if a patient finishes that long-term follow-up study, for us to clean, analyze, interpret, and publish this data. And this is done in collaboration with all study investigators. So at this time, we do not have an estimated timeline for that presentation or publication. But Ultragenyx is fully committed to both advancing the MPS IIIA research for the long term, and we are committed to sharing these research results with both scientific, regulatory, and importantly, the patient communities to enrich the collective understanding of MPS IIIA and advance care in this group of disorders. Next slide. So first, I want to address the transition of sponsorship from Abeona to Ultragenyx. And again, the nomenclature has changed, at least internally. It was formerly called ABO102, and now we're referring to the drug as UX111. Next slide. So here's a timeline of understanding the history that you heard from Glenn earlier. This was under development by Abeona, initially from Nationwide. The ABT-001 study is an open label at the time dose escalation trial that evolved essentially from a phase 1/2, which was a safety and dose finding study, into a phase 3 or registrational trial to confirm efficacy. And that's where that MITT group was created through a protocol amendment for that phase 3 portion. There was a separate study called ABT-003 that was in more advanced stages, patients with more advanced stages of MPS IIIA, and that was early terminated prior to this transfer. The data was presented, as I mentioned, from cohorts 1, 2, and 3 at the ASGCT in May 2022. So in May 17, 2022 is when Ultragenyx acquired the global rights to ABO102. And during this transition period, a lot of activities were ongoing. We provided close support and monitoring of the study sites, and we'll show you where they are located. In June 2022, the last patient was dosed with ABO102 or UX111, and the study was now fully enrolled. In November 2022, we issued an urgent safety measure to the sites to evaluate a potential immune response to the transgene product, which you'll hear more about. And in December 2022, importantly, in our LTFU, our long-term follow-up study, we amended the study to allow recruitment back into this long-term follow-up for patients who've completed the LTFU already, or from patients from the 003 study. So eligibility is basically any patient who had received UX111. And that was amended in December and then locally rolled out in the different sites. We assumed regulatory responsibilities, and our focus is on accelerated approval, which I'll speak about in a few slides. And most importantly, to be able to start manufacturing, and that started in this transition period, and you'll hear more about that too. So again, our data snapshot today is taking from August 16th, 2023. It's timed for when all 17 patients have reached at least 1 year of data after gene transfer. And this data is to support our regulatory path for accelerated approval. 2024, we are focused on that path forward. We're going to have ongoing interactions with regulatory agencies. You'll hear about the most recent Reagan-Udall Foundation workshop and that effort to align not only Ultragenyx, but other stakeholders in the community to educate and inform the FDA about the role of CSF heparan sulfate as a biomarker that could predict clinical outcomes. This was very important for our regulatory strategy. And of course, we're having ongoing analysis of data and reporting of results as appropriate. Next slide. So this is just a schematic of the clinical development program that I've mentioned already. Again, this will be recorded so you can look at this in more detail. But to date, a total of 33 patients have been treated with UX111. And in the ABT-001, this essentially phase 1, 2, 3 study, the focus of today's discussion is cohort 3, the highest dose, for which 22 patients received that dose, and 17 of whom have met the criteria of under 2 years old or over 2 and with a cognitive developmental quotient of greater than 60 or above. And there are 17 patients that define that group that we're focusing on today and their long-term data. And once again, this study, along with the 003 study, will then feed into the long-term follow-up, which we have extended that study to allow for continued follow-up. As of January of this year, 23 patients have been consented and 21 have already had 1 visit in the long-term follow-up under this protocol amendment. Thank you. Next slide. And this is just to show you, we had some questions about where our sites are located globally. So we have 2 sites in the United States, Columbus, Ohio, Nationwide Children's with the PI, Dr. Kevin Flanagan, Pittsburgh at UPMC with Dr. Rajan, and we have 2 sites in Spain, as you can see here, in Santiago and Barcelona, and then we have a site in Adelaide, Australia with Dr. Nick Smith. So Dr. Pico, Dr. Maria del Toro are our Spanish PIs. Next slide. So when we think about the drug development and the clinical trials process, as I've mentioned, ABT-001, that study has evolved over time. And really, this is a reference for you to look at offline. But as we develop from preclinical to clinical, our first phase of a study is typically first in human, is called a phase 1, or it could be combined in phase 1/2. And the focus of that is safety, and it typically involves a small number of participants and may not have too restrictive I&E criteria or inclusion-exclusion criteria. And then in the phase 2 to phase 3, we start to expand the number of patients. Sometimes studies are conducted separately, right? Oftentimes there's a phase 1, phase 1/2, and then a phase 3. But here what we're seeing is that this same study evolved over time. And that's why you may not see an official end to the phase 1/2 portion, because essentially the study is ongoing. And this will eventually then help us take that phase 3 study forward to the regulatory authorities to show the efficacy of our drug and the long-term safety. Okay, next slide. Oops. So it's really important to understand this regulatory strategy that we've been very vocal about in both in the public domain. So we are pursuing an accelerated approval pathway, and that is very critical to advance progress for rare disease treatments like UX111 for MPS IIIA and for other neuronopathic MPS disorders, and really and truly for other rare monogenic disorders. So what is this accelerated approval path? Well, this is actually put forth by the FDA, and it allows us to base our initial approval on a marker or biomarker. Here we're calling a surrogate marker that should reasonably likely predict clinical benefit. Now, a surrogate marker or biomarker or clinical marker is theoretically should be measured earlier in a trial, oftentimes before clinical endpoints become fully realized. And so the traditional approval path is typically longer, where we have to wait for clinical outcome measures. And so the accelerated pathway allows potentially earlier access to novel, safe, and effective therapies. So this is only really possible. This pathway is really possible only for conditions that are serious or life-threatening, and for where there's no other available therapies. And then in these diseases where we do have a candidate biomarker that is meaningful and related to the underlying disease state. And so we feel strongly that there's enough preclinical and clinical data connecting a biomarker to clinical outcomes, and you'll hear more about that. This biomarker is called heparan sulfate, and it's measured in the CSF. However, so even though we might gain initial approval under accelerated approval, this needs to be followed up with long-term confirmatory studies, either the same study or additional studies, to verify the clinical benefit, right? So we have biochemical efficacy that gives us initial approval, but we have to come back years later to say to the FDA, we do have a positive effect on clinical outcomes, and those clinical outcomes are defined or in partnership with the FDA. However, what's really important is that if that clinical benefit is not verified, the FDA can seek withdrawal of that therapy from the market. So that's why it's conditional. It's not a full approval. And that's what we are pursuing right now. Next slide. Okay. So what I want to talk about, based on some of the questions that were posed to us from the community, we want to talk about gene therapy for MPS IIIA, but also concepts that are common to a variety of gene therapy that's in development for other disease states. And there's common themes with in vivo gene therapy. Next slide. So I don't need to tell this community specifically what Sanfilippo is, but I do want to use this slide to highlight the biomarker that we're measuring. So as you know, right, Sanfilippo type 3A is a specific genetic condition, unique from biochemically from 3A, B, C, and D. In this case, the condition lacks sulfamidase, which is the enzyme needed to break down a really important sugar called heparan sulfate. And all 4 subtypes of Sanfilippo are involved in that breakdown, but each has their own unique enzyme. The lack of this enzyme results in accumulation of this sugar in brain cells, and this is detrimental to a brain cell's health and function, and it progressively leads to dysfunction and eventual brain cell death, which you can actually see on MRI as shrinkage of the volumes over time. Clinically, we know what happens. This ultimately leads to loss of previously acquired skills such as language, cognition, and motor, and most importantly, early death. So this is the biomarker that we're going to be speaking about later in the presentation. Next slide. I also want to highlight the importance and value of looking to the published literature and speaking to those who have created natural history studies. This natural history data has helped researchers be able to detect when there's a change between treated and untreated patients. And this is the Shapiro study that was published in 2016 and is still very critical today. The only thing I want to highlight here is this is helping us educate and discuss with the FDA the expectations of a therapy. In those first couple of years of life, children might be developing with relative positive developmental slope, meaning that they're gaining skills. They might be gaining at a rate of a neurotypical child. However, then there is this turn where there's a plateau where that slope is starting to become 0. And then there's a regression or loss of skills over time. And this triphasic course is very important for when we're trying to interpret our data on our patients and how they're performing after treatment with 111. So I just using this as a comparator for later. Next slide. So what are these key issues and challenges in developing a treatment for MPS IIIA? MPS IIIA is likely underdiagnosed and misdiagnosed globally. As I said, and as you know, as the patient community, sometimes these clinical symptoms begin after a period of neurotypical development. And sometimes the developmental delay or the features of the disease might be misdiagnosed for general global delay or autism spectrum, and it doesn't lead to a confirmed diagnosis, and there's a lag in the understanding of the genetic basis of that child's developmental issues. There's a small patient population compared to other diseases out there, and that's spread across the world. And we don't have validated measures for clinical outcomes. We do have clinical assessments used to detect motor, language, and cognitive development, but they're not specific for MPS IIIA. And this has implications for identifying patients, recruiting patients, and enrolling. Now, the age of patients is an important topic, especially for a disorder that affects the brain. Early diagnosis is critical to allow for early intervention to maximize that benefit. It's really in that early developmental period, children are learning at rapid rates in neurotypical development. But with a disease that has onset in this early childhood period, children might be impacted differently and to different degrees. And this also impacts our ability to identify and recruit patients. And it could take longer. And that leads to delays in recruitment and prolongs the study. Next slide. So again, going back to the type of gene therapy we're talking about for UX111 as a reminder to the community, this is a in vivo AAV gene replacement therapy. So each of those terms are very important because there are other types of gene therapy out there that might be in development for other disorders. So the gene replacement therapy aims to treat the disease by directly transferring that corrective genetic material to a patient. So the gene that encodes sulfamidase, that protein, has an error in it, and we're trying to give back the correct sequence to the patient. And we do this by delivering it in a modified virus that cannot replicate or cause disease. We take out those pathogenic or the bad elements of that viral vector, and we put in the correct gene inside of it. That's the cargo. And that virus then inherently will infect the body and deliver a functioning gene that will then be translated into a protein. So very complicated, but in essence, we are replacing back that enzyme, by replacing it with a corrective gene. And so we're hoping that we can target those affected cells with those viral vectors to potentially alleviate some or all symptoms of the disease. Okay, next slide. So what are the benefits, or what do we know about AAV gene therapy? So the AAV is the specific type of viral vector, of which there are many different serotypes. In 111, we're using AAV9, but AAV in general is well studied and an established vector backed by 50 years of learning, going back to the 70s. So we understand a lot about AAV, but we still have to learn more about its application to the specific disease state. And there are many versions or serotypes of that vector that's been studied in animals and in humans. So we have robust testing as of today. Now, AAV gene therapy can target non-dividing cells, such as the cells in the nervous system and eye, but it can also bring along with it immune responses, and that can be to the particular capsid of the viral vector, or it could be to the transgene itself. But the AAVs are engineered to replace the viral genes with this therapeutic gene, as I mentioned before, and they're not to integrate into the patient's DNA. And what does that mean? It means that it goes inside the cell, but it sits alongside the patient's own genome. So we typically don't expect any integration or disruption of that patient's own genome. So it's non-integrating. So it's really so how does this affect our ability to deliver this therapy? Well, although this AAV is a benign virus that doesn't cause disease, many people have had prior exposure through natural infection, and they might have pre-existing antibodies. So you'll see that patients with select AAV antibodies may be unable to receive that therapy because that means that they could potentially develop an immune response to that vector. And those are definitions below of the vector and the capsids for more reference. Next slide. So more about the immune response. This is a really important area of gene therapy that we are learning more and more each day and each year. And as stated, this immune response is a normal defense mechanism of the human body. The human body is designed to attack external or invading viruses. And that immune response can vary from person to person. And so although we remove a lot of the pathogenic parts of a viral vector, it still recognizes non-self. So the immune system can generate antibodies to that vector. That's the capsid on the outside, or actually to the product that comes from the transgene, in this case, the sulfamidase enzyme. So you'll see that Ultragenyx and other companies will test a patient's blood for pre-existing antibodies to that vector capsid prior to administration of gene therapy. And as you can see on clinicaltrials.gov, in this trial and other trials, it is part of the inclusion-exclusion criteria, meaning if a patient has elevated antibodies to that particular serotype or that particular virus, they are excluded from receiving that gene therapy. So why is that? That's because the immune response can impact both efficacy and safety of that gene therapy. And our goal in patients is first, do no harm, right? We don't want to harm a patient, even when the disease itself is so severe. We are trying to understand that more, and that's why we have measurements to understand this impact. So here, how does it reduce efficacy? Well, the immune cells, if they exist, pre-exist before, they can attack the virus as it's trying to enter into the target cells, as it's trying to enter the brain. And if it gets sucked up or attacked before it gets inside the cell, then we can't deliver that corrective gene, potentially reducing the efficacy of that therapy. So that's efficacy. Safety-wise, it can cause a massive immune reaction and inflammation, and this could potentially cause cell and organ damage. Now, all of these are now known because we've been studying these viruses, and there are ways to mitigate or reduce that risk of harm, and that's by excluding patients with elevated antibodies at this date, as well as providing immunosuppressant medications proactively or reactively. Now, over time, we've seen a change in the prophylactic or preventive immunosuppressant medications from different programs. In UX111, they were initially given steroids, a short course. In this, when we saw the immune response to the transgene product, we then incorporated additional immunosuppressive medications that could be used if warranted. And we'll talk a little bit more about that. Only one patient to date has underwent that. And this is to help reduce the immune reaction, inflammation, and hopefully prolong efficacy. But these immunosuppressive medications have their own risks and potential side effects. And it's really important for families to understand this and gather their input about those additional medications before we administer them. And that's why they're given under consent. Next slide. So how do we, what are these overall challenges? I'll just briefly go through. Finding that right dose is very important in those early phases of the study, phase 1 and 2. I talked a little bit about the inclusion-exclusion criteria that can be very impactful if it results in exclusion of a child from a trial due to those pre-existing antibodies. The treatment goals have evolved over time. People used to call gene therapy curative, and I don't like to use that term because we're not clear that we can reverse all of the symptoms. We do hope that we can impact the natural history of the disease and provide benefit, but it's not clear that we can reverse all of those symptoms. We call it one and done. We like to think that we can give it once and that it's durable over time, but we don't know, and that's why we need to follow it up over the long term. So we hope to understand that response to gene therapy. We need to understand if age has an impact on the degree of clinical benefit or the type of clinical benefit. Patients may still require supportive care and therapies even though they receive gene therapy. It might limit their ability to participate in future treatments. And this is really important when you're consenting a patient for gene therapy, because if there's other therapies down the road, patients may or may not be eligible if they've already received gene therapy. And so that's really important. Other considerations at this point in time, we are not able to redose a gene therapy, and there's a benefit risk to that. Finally, long-term effects. As I mentioned before, it's really important to understand the full impact of a therapy. And if we might have immediate changes in clinical outcomes or in biochemical outcomes, but truly we want to know what happens over the long term as that child grows and develops. What are the long-term efficacy and safety? And that's why typically for in vivo gene therapy, we're following patients for years, 5 to 10 years, sometimes longer. To understand that, and it really requires active participation from the patients, families, and doctors. Finally, we could have a whole lecture on manufacturing, but gene therapies require a very high level of accuracy and consistency when we're scaling up from just small batches to larger batches to be able to provide this therapy to a community. And so this is a very resource-intensive process, and it takes a long time to develop and ensure that we have a high-quality product. And we're in the midst of that now. Okay, next slide. So as a reminder about UX111 specifically, as I stated, it's a gene replacement therapy to target that underlying deficiency of sulfamidase enzyme to hopefully clear out those toxic levels of heparan sulfate, that sugar. So UX111, formerly ABO102, is an in vivo gene therapy with a full-length copy of that sulfamidase transgene. It's administered intravenously, and it's being developed for treatment for children with MPS IIIA. So we hopefully will directly transduce brain cells as well as provide an additional source by cells secreting it in the surrounding. Again, this is for your reference. Next slide. Okay, I'm going to pause for a second. Matthew?
Hi there. So question submitted that bears upon the presentation so far, and these involve questions on dosing and redosing. Is there a potential to redose a patient with UX111 if they've received it in the clinical trial?
Thank you, Matthew. So finding a path forward for patients who test positive for these antibodies is a continued interest for Ultragenyx, as well as for many developers in this gene therapy space. However, at this moment, the UX111 infusion is a one-time only infusion given to clinical trial participants who are negative for AAV9 antibodies.
And one additional question along these lines, can my child potentially qualify if they've gotten ERT before?
So ERT or enzyme replacement therapy, right?
Yep.
So currently our UX111 studies are fully enrolled and we're not accepting any more patients. So I can't really comment about currently. Looking ahead, it is premature for us to talk about how patients may be able to access UX111 outside of a clinical trial as well. But our intent is to gain FDA approval of UX111 for patients who may benefit.
Thank you.
You're welcome. Okay, next slide. All right, so we're going to give highlights from my World Symposium presentation about our recent data snapshot. Next slide. So this data presented at World was also presented in another form at the Reagan-Udall workshop. This workshop brought together academics, other sponsors, and patient advocacy groups to educate and talk with the FDA about using CSF heparan sulfate for a variety of programs in the neuronopathic MPS space. And so I'm presenting similar data from both of these presentations there. So they are publicly shared, and we're showing for the first time in this study a correlation between reducing that biomarker, HS, and longer-term clinical outcomes, which are measured by the Bayley Cognitive Raw Score. So again, this data snapshot is just focused on that target population within cohort 3. So those youngest under 2 or over 2 with a developmental quotient of 60 or above. And this is pretty much our phase 3 portion of our study, and that we're moving forward with for accelerated approval. So as of this snapshot, all 17 patients in this group reached at least month 12 of their month 12 post-gene administration. 15 of those 17s were at least 30 months of age. So age is important to understand how these children are doing over time. And 6 of the 17 have reached at least 5 years old. And the mean study duration was 28 months with a range from anywhere from 11 to 60 months. Okay, that's the background. Next slide. So the baseline demographics really just is describing this population. So a younger population because of that criteria, slightly more males, and the baseline CSF-HSs were elevated to over 300. This is a mean value. And here you're seeing the Bayley Cognitive Raw scores at baseline were about 47, and the DQ was 85 in 13 of our patients. Next slide. Okay, so what I'm going to show you first is our biomarker data, right? Our biochemical response to UX111 in this target population. On the left side is our heparan sulfate, or HS. This is just mean % change from baseline. And you can see that the direction downward is a reduction. And we see that at 1 month and then a further reduction by 6 months. And as you see, the N, or the number of patients data, is on the y-axis. So by month 12, we have all 17 patients data. At month 24, we only have 8 patients data, and we see a slight rebound, which is heralding, or in some of those patients, a rise. It's associated with a rise in antibodies to the sulfamidase enzyme. But then overall, we're seeing a sustained reduction at least 50% or better. This, again, is group level. This is not individual patient data, but it's important to understand that we are having an impact on the CSF's accumulation of HS. Now, when we look at exposure over time, meaning we're looking at all the values after baseline, called area under the curve, we actually see 63% reduction. That's really quantifying how much impact we're having, hopefully, on the brain. So what's the other side? The other side is gangliosides. These are also important metabolites that are inside the lysosome that accumulate independent of the sulfamidase enzyme, but they're really reflecting that the lysosome, or the brain cells organelle, is functioning better over time, and it's also going down over time and stabilizing. Next slide. Okay, so that's the biomarker data. Now, as we follow our patients forward, we are continuously assessing their cognitive outcomes. Here, we're only showing cognition because that's our primary functional endpoint. But I would like to highlight that we are collecting language scores and other scores in activities of daily living and other measures outside of cognition. But what we were asked to show is our effect on cognition by the FDA. In green is our treated cohort of patients of 17 patients. In blue are the natural history from that Shapiro study that I showed you earlier. And once again, we can't reliably detect differences in all our patients just yet, but we're starting to see a trend or a divergence over time. Now, the first 2 years of life, both groups are gaining in their Bayley Cognitive Raw Score on the y-axis. So that means they're gaining skills. And so it's really hard to see a treatment effect to say that the 111 is having an effect or not. We cannot reliably say that. However, as we start to see the patients over time reach 4 years and then 5 years old, we're starting to see a separation in the two groups, meaning that our patients are continuing to gain in skills while the untreated cohort has lost skills over time. And at month 60, there's a 23-point mean increase in the treatment group over the untreated. And this is statistically significant. Now, there are a couple of patients that are losing skills in the treated cohort. And I want to highlight that here. So, a therapy, we want a therapy to be 100% effective. But that's not necessarily needed for approval. But what's important is that to us, when we see a loss of efficacy, it led us to look further. And that one patient that's going down on the slide in green is a patient that developed a significant high sustained anti-sulfamidase immune response. And that actually, if you look back, you can see that take hold with a biochemical rebound of heparan sulfate 1 year prior to loss of cognitive improvements. So what does that mean? It means that the immune response was mounted. It led to the cells to reaccumulate the heparan sulfate, and those cells eventually started to not function well again. And so that child who was previously improving has started to decline. And now that led us to issue that safety message, not so much as safety, but it's more of an immune response about efficacy. And that allowed me and others to alert the sites to evaluate patients if needed for this immune response and treat if needed, sorry. And so to date, we've evaluated many patients for this immune response, but one patient is now currently treated with additional immunosuppressive medication because they developed high titers and a rebound of the heparan sulfate. So they reaccumulated that. But the child has not yet shown cognitive decline. So we're trying to intervene before, because our hypothesis is that the heparan sulfate reaccumulation will lead to later cognitive decline. Just like we think if you reduce heparan sulfate, it could lead to cognitive improvement. And so that patient is being treated right now, and the response is currently under review. We don't know the full answer yet. So, but for full disclosure, this is been alerted to all the sites and incorporated into their amendments. Okay, next slide. All right, so now it's important to connect the dots, right? So the FDA wants to understand, is this biomarker, heparan sulfate, reasonably likely to predict clinical outcomes? Is there a connection between the two? You reduce one and you improve the other. And so we are showing here that there is a relationship that's statistically significant. It's a little complicated, but this figure here on the left, the graph, shows that on the y-axis, children gaining estimated yearly change of positive gain in cognitive raw scores, cognitive skills over time. And then there's a reduction of CSF exposure. As you can see, it gets more and more negative. 15 of our 17 patients simultaneously achieved both a reduction in that heparan sulfate and a positive gain in their Bayley cognitive scores. And this correlation is statistically significant when you do the Spearman rank order correlation coefficient. And it's maintained even when you adjust for age. So this is very complicated, but we needed to show that there is a connection between reducing heparan sulfate in the CSF and later cognitive benefit in order to make that case that HS is potentially a biomarker reasonably likely to be predictive. Okay, so very complicated, but important to show that in the majority of our patients, we're showing a correlation. Next slide. Okay, so we also looked at brain volumes. Now, brain volumes are important pictures of the brain to show how the brain cells are doing. It's a very gross or big picture. Large, gross is large in medical terms. So we're comparing it to the natural history data. So in black are our patients. They have larger volumes initially, likely due to storage, and then they decrease a little bit and stabilize. So removal of storage and then it stabilizes over time. But what we see is the pink patients are going down. So there's volume loss or shrinkage of the brain over time in untreated patients. Now, it's hard to say for sure what's happening with our patients because the data is still only a few years old. So brain MRIs are a longer term marker, right? It's not an early marker like heparan sulfate, it's a longer term marker, and we have to follow them over time. We're incorporating a brain MRI in the long-term follow-up study at the recent amendment. So again, this is helpful, but not the full story yet. Next. Safety. So any part of the, anytime we present about our program, we should really focus on what we've learned from a safety perspective. There is standard language used throughout all different clinical trials, but how to capture safety. And these are the definitions on the slide. Again, you can refer back to it, but we'll talk about adverse events as any untoward medical occurrence that occurs in a patient during a clinical trial that's received an investigational drug. It doesn't necessarily have to be caused by it. It just has to happen after the patient has received that drug. And then it's up to the principal investigator to assess whether it's related or not to the medication administered, in this case, gene therapy. But we have to report out all adverse events. And so the treatment emergent ones, again, after the medical treatment is delivered, it emerges, it may or may not be related. Then we have to categorize it. How serious is it? And what's the severity? And a serious adverse event is one that results in either death, it might be life-threatening condition, a hospitalization, permanent disability, birth defect, or there's a basket group here of identified by the principal investigators, just simply a very important medical event. So really, it gives a lot of power to the PI to assess the connection, because they're the ones at the bedside seeing the patients after the treatment. And then we grade it, mild, moderate, severe, life-threatening, and grade 5 is death. Next slide. So when we think about the ABT-001 study, which is that first 2 years post-gene therapy, we've seen no treatment emergent adverse events that led to study discontinuation or death. There have been no so none of those in this trial. The most frequently reported, though, are related to elevations in liver enzymes, and this is the majority of these were mild or moderate, and they resolved on their own, and that's important. We do understand that in AAV gene therapy that's delivered systemically and in other forms, we do see liver enzyme elevation. So it's not uncommon. It's a known side effect of AAV gene therapy. We did have one event that was grade 3 that was a specific enzyme of the liver called alanine aminotransferase. It elevated high enough to reach a grade 3, but thankfully it resolved. So again, it kind of went up and down. Most of these liver enzyme bumps are related to the immune effects of the capsid. So it's a very well-known class effect of gene therapy. Next slide. So overall, we've talked about the data that we presented as interim data from a target population of patients in cohort 3. And we've showed a reduction in that CSF exposure over time. We showed other reduction in other types of biomarkers in the cell that are important. We are starting to see stabilization of the brain MRI volumes, and they were within normal limits. And overall, the safety profile, there's a favorable benefit risk profile of UX111. Again, these are interim results as of today, as of August 2023. Thank you. Next slide. Just want to thank at this point our principal investigators around the world, my colleagues, and of course, all the patients and caregivers that are participating in this trial, because this is really critical for not only for UX111, but for MPS in general to understand the potential of gene therapy. Okay, next slide. Okay, back to you.
Okay, thank you, Dr. Lau. We have some questions around the efficacy portion of your talk, and I know you answered quite a number of those, and we actually really only saw the data for that cohort 3, or that kind of phase 3 youngest group of children. But one of the questions was about the effectiveness and how age plays into that, if you can comment at all.
Yeah, so the residual symptoms in the youngest treated, I'll tackle that one first. So we do not know fully what residual symptoms may continue to exist or develop over time, right? So we need to follow these patients long-term to measure the full potential benefit of UX111 in the context of ongoing development. And so we are conducting that long-term follow-up study to monitor all patients who have received UX111 for a minimum of 5 years post-treatment. Most of the youngest children in this study are those in cohort 3 by the nature of the inclusion-exclusion criteria, and they received that highest dose. And they were also the last participants. Most of them were the last to enroll. So we only have the first year or just over a year of data in those patients under this newer inclusion-exclusion criteria. So it'll be really important to follow those younger patients over the long term. So it's evolving over time, and we want to understand both efficacy and safety in years to come. So how important is age in administering the drug, and does the rule younger the better apply? Again, to talk about the natural progression of MPS IIIA, we understand that children might begin to slow in early childhood in their skill acquisition and then regress later after that initial period of gaining skills. It's not known, it's unknown whether we can regain skills once they're lost and to what degree. And that is why we want to follow up our patients who received 111 in the long-term follow-up. However, in general, it would be optimal to intervene with gene therapy before there's irreversible brain damage. We may see a greater therapeutic response before this irreversible damage occurs. However, we do not want to forget the significance of possibly stabilizing a fatal disease or helping to stop it from progressing further in other patients. And so, for example, a child with MPS III is already significantly regressing. There still may be benefit in receiving gene therapy in that it might help them stabilize. Okay.
Yeah. Great, thank you. And then the other questions are really around some of these non-cognitive effects that you may be seeing, and how are you actually capturing those in your clinical trial? Those may be things that, related to, as you refer to, ADLs, like, walking, swallowing, language, which is, much more of a cognitive aspect, but other ways of communicating, impact on the retina, hearing, and all of these other kinds of ways that the treatment may or may not have an effect.
So while treatment effects on cognition was one area that you clearly show that we are assessing, there are other domains such as the communication and motor skills that we're currently reviewing that data now, and we understand that these are meaningful to the families. So as well as the patient's participation in activities of daily living and their adaptive behavior are being explored and not yet ready for presentation. Now, specifically in the age 2 to 3, the last data presented was in May 2022, and that did encompass the cohorts 1 and 2 and 3, and it included, that was prior to our licensing. Although we are focused now on the phase 3, we will review all this data in totality. So that data presentation had information from the Baileys, the Vineland, the Mullen, and cognitive functions. And we would like to circle back to that and understand fully how that's developed over time, but we don't have a timeline for that. So we're in the midst of reviewing all the data, and we'll update the data from cohorts 1 and 2 as there's longer term data. The standard process, I think Kristen had already mentioned, we will report results out for a study after the last patient in. So for 1 year after the last patient in, we will be reporting the results of the pivotal transfer A and the long-term follow-up studies. We're committed on that to clinicaltrials.gov. Those endpoints will include heparan sulfate, neurodevelopmental outcomes, as I mentioned, the gangliosides and brain volumes. But any other study drug effects or insights will be reported once the studies are completed as part of that full data analysis. We're not clear on everything. But those last combined questions, I think, Cara, is that what you were asking as well?
Yeah.
Yeah, so it's hard to directly comment. So any other study effects, drug effects, or insights beyond what were shared today and throughout our February data presentation will be reported once we complete a full data analysis. And that's what I can say about that at this point.
Okay. Okay. Thank you. Matthew has the next group of questions. So.
Thank you. So these questions are around certain aspects of the trial associated with safety. In particular, the cohort 1 and 2 were lower doses as part of the dose escalation. What kind of learnings came from these particular aspects of the study?
Right, thank you. So as it was originally designed as a phase 1/2, these cohorts really contributed to our understanding of the initial safety that allowed us to dose escalate. So based on the safety and efficacy of first cohort 1, then 2, we could go up to that highest dose to that highest dose. In the future, we do want to review the long-term follow-up data obtained for those children in those cohorts. And that actually might give us more insight to an understanding of that long-term safety and efficacy. But it did really inform that initial ability to go forward. It does take time to see full impact, and so both the short-term and long-term effects are important to understand from a safety and from an efficacy, as I've mentioned. And this is applicable to all cohorts, even the, quote, lower doses. And as you know, because MPS IIIA is neurodegenerative and slowly, well, relatively slowly progressive in the eyes of other rapid ones, it's still progressive. And we need to follow over time to understand the full impact, and that's what we're doing.
So the next 3 questions really revolve around issues of age and size at treatment. In particular, in an older or heavier child, the viral dose is going to be higher. What are the consequences there? Also, what is the lowest age where we can see administration? And is it theoretically possible to treat a fetus prenatally with this sort of gene therapy?
These are all really good questions. So when we inherited the program, at that time of transfer, there was no upper weight limit restriction. And so the previous dosing strategy was really informed by the program's non-clinical data, and there was no restriction imparted. So as with other types of phase 1/2 studies, that primary goal was to understand safety of that dosing and those various doses. And we dosed for weight, so adjusted for weight. There were no significant safety findings to date based on those receiving therapy so far. For the lowest age of administration, so based again on the program's non-clinical data, that informs the design and the ability to dose patients. This program was approved for administration, approved by regulatory authorities around the world to administer from birth through all pediatric ages in some countries. There are age restrictions that, in certain countries due to different decisions by the health authorities. So that's by the health authorities' decision, not by our program, which did impact the age of the patient. But essentially, in certain regulatory environments, our intention is birth through all pediatric ages. For the fetus, so Ultragenyx has not explored developing gene therapy to a fetus at this time, and we do not have any data to support that endeavor.
So the final question is really around shedding of the virus, which happens to some degree after it is administered. And this is not virus that is reproduced. This is just the vector that's leaked out. Is that a concern for other children in the household? They could potentially be exposed and mount antibodies. So how long of that period where the treated child may need to be isolated?
So viral shedding is a standard assessment for in vivo gene therapy. And each program, even if we use the same viral vector, is required to develop a shedding profile and measure this. And this data is currently under analysis. There is a potential for exposure in families treated with AAV, and a family needs to discuss that, any questions and necessary precautions with the child's treating physician if they're participating.
Excellent. Thank you very much.
Thank you. Next slide.
Okay, now on to the future of the program. And as Kristen mentioned, this is where the bulk of the questions came in, and really to be kind of summed up as what's next, and really questions around when will access be available, in what ways, including when the drug supply becomes available, expanded access, commercial program, all of the above.
Sure. Okay. Well, I mean, obviously this is very important, right? And this is why we're here to communicate to you. So our goal, first and foremost, our goal is to make UX111 available to the MPS IIIA community as soon as possible. And in order to do that, we must focus on working with regulators on a path to approval. And to that end, our objective, as I stated before, is to pursue an accelerated approval pathway in the United States, using CSF-HS as a biomarker that measures the underlying disease. This will not only support a faster path to market for our drug, but overall innovation in the space for other neuronopathic MPS. And as a result, we believe that future clinical studies that use a biomarker instead of clinical endpoints to show efficacy would be easier to enroll and much faster to complete. We're trying to make change for the community. So while we're in discussion with the regulators, we do not yet have a timeline to share on when we expect to file a regulatory submission for marketing authorization. It would be premature for us to talk about these topics, such as when and how patients may be able to access 111 outside of a clinical trial. But in tandem with regulatory discussions, we are ramping up manufacturing of UX111, which is critical to ensuring future access to the drug. We recently selected a manufacturing partner and have successfully transferred the manufacturing processes to their facility to develop the batches required for future regulatory filings. We recognize the community's genuine interest in the future of this program, and we're committed to providing the MPS III community with updates, with regular updates, and we will share additional information when it is available. To address the major challenges, I have a separate so the major challenges to getting approval. One common challenge across many programs in the MPS disorders is that the FDA historically required demonstration of efficacy based on long-term clinical endpoints like cognition. This is particularly hard to do in an ultra-rare condition like MPS IIIA, where children are not diagnosed, they're diagnosed well into a state of neurocognitive decline. And so achieving a statistically significant difference between treated and untreated patients in studies requires enrolling tens of patients, that only enroll tens of patients, is problematic because it might take years to demonstrate. So meanwhile, we have strong data today showing that treatment with 111 results in reduction of CSF-HS, which is a well-understood biomarker and underlying disease. We still are having difficulty with that path forward. So with UX111, we are now measuring we are continuing to measure neurodevelopmental outcomes. And we're working to demonstrate that there's a connection, as I showed today, between reduction of HS is reasonably likely to predict that clinical benefit. And that is the requirement from the FDA. This is critical because if we do establish that HS as a measure of that, if we can establish CSF-HS as a measure of the underlying disease, and that it's, and when you evaluate it earlier in a trial than a traditional clinical outcome measure, it would enable us to pursue that accelerated approval pathway. So it's really critical. You know, a biomarker is not relevant necessarily to the particular patient. It is a path, it's a regulatory strategy to path to access, right? So we recently co-sponsored this pre-competitive workshop that I talked about, hosted by the Reagan-Udall Foundation, entitled Qualifying Biomarkers to Support Rare Disease Regulatory Pathways, and the case example of heparan sulfate in neuronopathic lysosomal storage diseases. We worked with this foundation to bring together the researchers, the clinicians, patient advocates, and other biotech companies working in this space to share a body of evidence with the FDA supporting that HS is a biomarker that should be qualified to support access through the accelerated approval pathway. Okay, and your final question here about drug supply. So in December 2023, Ultragenyx announced that we successfully transferred the UX111 good manufacturing process to Andelin Biosciences. Andelin is the Contract Development and Manufacturing Organization, or CDMO, spinoff of Nationwide Children's Hospital and the original drug product for UX111, formerly known as ABO102. Which was originally manufactured at Nationwide. Getting that manufacturing process set up was critical for any future regulatory filings. We had to show consistency between the clinical and commercial product is really important from that regulatory perspective. So we are partnering with Amylin to help move us forward. So that's a whole other aspect of that pathway that is important to understand to the community. I personally am learning about it too as a clinical person, the manufacturing part. So that's that for the future.
Yeah, thank you, Dr. Lau. I just wanted to clarify one piece in case I might have missed it, but the aspect of, if you can comment about further trial opportunities or opening more opportunities within trial. I know you can't comment on access outside of trials at this time, but are there any kind of plans for more access under a clinical trial setting?
We can't comment on that at this moment, but as soon as we have an update, I will absolutely be communicating with you and with MPS Society to get the word out.
Yeah, great. Thank you. Matthew, back to you.
Sure. So some additional questions. The modified intent to treat group is a very young group of patients. How did these happen to be recruited and enrolled? Did they have an older sibling or a family history?
Right. Thank you for that question. It's a really important understanding. So Ultragenyx was not we're not involved in that initial designing of the protocol that included that group. And we were not involved directly with the recruitment or enrollment of this cohort. So that's the caveat here. The cohort had already been enrolled and almost all patients were dosed, with exception of one patient by that time we acquired the program. But typically, study sites and study investigators are responsible for determining which patients are eligible and identifying and which are best suited for a clinical trial. It's a decision between the study investigator and the family. Sponsors do not take part in those decisions. We do recognize, though, it's incredibly difficult to diagnose MPS III so early. And it's often the case that new patients are identified at younger ages through genetic screenings or the diagnosis of an older sibling. We can speculate this may be the case for the MITT recruitment. However, we were not directly involved with that program at the time and cannot speak to this.
Excellent. Are there learnings and applications from the data derived from UX111 to MPS IIIB?
So the ABO102, now 111, is focused only on MPS IIIA. We cannot comment on other programs, including any Aviana programs for Sanfilippo B. We understand the urgency for treatment in all types of Sanfilippo. While there are trials that have been studied for MPS III, it's best not to extrapolate our data to theirs. You may consider contacting organizations such as the Sanfilippo Foundation or the National MPS Society or your child's physician for more information.
And other gene therapy research and or trials that the community may have heard about that are potentially coming in the future. How is the UX111 program different than the product that is said to be under development at University of North Carolina?
Yeah, it's great that the community is following drug development so closely, as you should. However, Ultragenyx cannot comment on another program. We recommend you contact organizations such as Sanfilippo or the National MPS Society or your child's physician for more information.
Certainly. Thank you so much.
Thank you. Okay. Well, Kristen, I just want to thank you. A sincere thank you to all the patients and families that have participated in the UX111 studies, to the sites, the study staff to help conduct these studies and are continuing to work hard to follow these patients, to follow and clean and get that data to us so we can understand it. And I really want to say thank you for all of your time and attention today and those thoughtful questions that came across. And thank you to both Sanfilippo Foundation and the National MPS Society. Kristen, you want to say a word or two?
Yeah, thanks, Heather. And thank you, Cara and Matthew, for for hosting and doing such a great job at getting the community to submit questions and pass them on to us. I just want to echo a few things that Heather shared. We recognize that this program has a long history, and the community is very interested in a number of topics and research areas. And I think that really shows in the 60+ questions we received. And so we're very committed to providing you with more information when we have it. And as Heather shared, this is interim data at this point. And as the team is able to clean more data, interpret that, analyze it, prepare that, get it published, potentially put it out at congresses, et cetera, we are committed to finding ways to sharing that with you. And one of the best ways in doing that is through our partnerships with organizations like today. We recognize that, there have been 33 patients, and of course their families and caregivers and siblings who are impacted by this. And it's a long journey, and we really sincerely appreciate your generosity in advancing research. This would not be possible without you. And the more data that we're able to collect through the long-term follow-up, as Heather mentioned, the better able we'll be prepared to answer these additional questions about cognitive outcomes and about what other ways is UX111 impacting a patient and their future. And again, just to reiterate, because of the design of the phase 1/2 into a phase 3, as Heather mentioned, once the phase 3 portion has been completed and the last patient has had their last visit, following that, about a year later, we'll be able to put together a plain language summary of the clinical trial results, and that will be made available to the families in the study directly through the physicians, as well as on our website. So today we really view as just one time point in a conversation with you all, and we know that there will be more information to share in the future. So with that, we wanted to just share a couple additional resources. Heather did a really great job of sharing some background about gene therapy, AAV, in vivo, gene replacement, what are some common challenges. And I know that both the National MPS Society and Cure Sanfilippo and many others around the world are able to provide education about this. In case you're interested, the American Society for Gene and Cell Therapy is also a great resource. And so this is their website here in the United States. And then finally, you can find us online in terms of Ultragenyx in a variety of ways. So LinkedIn, of course, and Facebook. We also have a dedicated patient advocacy newsletter where you can sign up and learn more. So it's ultrarareadvocacy.com. And Heather and I and our team will be at the International MPS Symposium next week in Germany. I don't know if any of the families will be there, but we really appreciate your time and attention today. And we look forward to sharing more information with you in the future. Thank you. Cara and Matthew, I'll stop sharing and pass it back over to you.
Okay, thank you. Let me move back over here and go to our next slide. Okay, just a couple of quick wrap-ups. You guys did a wonderful job and shared so much information directly with our community, and we truly appreciate you all from the Ultragenyx team for making that a priority. As you said, there will be many more questions along the way as we go, and here are some ways where people can reach out and share their questions, either with Ultragenyx directly or through our organizations at Cure Sanfilippo and the MPS Society. And just to echo that, we are thinking of all the families out there who have not only been able to participate in this study, but those who are waiting and hoping to have access to something that will help their children. So Godspeed to everybody who is working hard on this and many other pathways to access. Matthew, say anything?
All of that, exactly. Godspeed to all, and thank you for tuning in. And please be in touch with us for any updates or additional questions.
Thank you all. And we will share the recording afterwards. Everybody have a wonderful day.